Now, karyokinesis occurs first step as prophase. In prophase, what we see, this is the cell, this is the entire thing is the cytoplasm, this green color is your nuclear membrane, and these blue and red are your chromosomes or DNA material. Now, this DNA material is generally invisible to our naked eyes, but during this M phase of the prophase, we have this DNA material gets condensed into chromosomes or chromatids. Okay, now condensation of the DNA begins. Centrioles begins to move to the opposite poles. Okay, now see, these two green color centrioles are moving into the opposite poles, trying to move into the opposite poles. Here we cannot see the centrioles at the opposite poles, they are moving. So the centriole duplicate in the S phase. DNA as well as centriole duplication took place in the S phase. We have studied it. Okay, now see, they are moving into the opposite poles. They have not yet. Now in the previous diagram, we have seen the chromosomes getting condensed and forming the chromatids. They have not fully got condensed. So in this, the phase, you can say late prophase, all the cell organelles have disappeared. The nuclear membrane have disappeared. Now these chromosomes or the chromatids, they are moving into the opposite poles. These centrioles have gone into the opposite poles. They have generated these asters. These are the microtubules. These are the asters, and they are connected into the kinetochores. See, what is the difference? Now this blue color inside is your centromere that connects to chromatids or chromosomes, and this green color lining is your kinetochore, to which these microtubules or the asters get attached. Okay, now this is your transition to metaphase. See how the chromosomes are slowly and steadily moving into this middle, into this equatorial line. Okay, now in metaphase we see they have moved into this equatorial line or into this middle plate, forming the metaphasic plate, and this is the most important significant point in the metaphase. Now see, these asters are formed star-like structure in the centrosomes or the centrioles, and they are radiating these microtubules, and now they are trying to now see, in the metaphase, what we see, well compact form of chromosome is seen in this metaphase. Now the study of chromosome or chromosomal study or study of any defect in the chromosome we do in this metaphasic plate. Okay, now spindle fibers are attached to the kinetochore of the chromosome. I have said chromosomes move towards the middle, forming the metaphasic plate or metaphase plate. See how they are forming the metaphase plate, and the kinetochores, they are connected to the centrioles, are now present at the opposite poles. Spindle fibers, asters, centrioles, they form a mitotic apparatus or apparatus. Okay, now coming to the next phase, anaphase. In this case, the chromatids separate and the centromere split. See how now we have this type of arrangement. Now they will pull this portion of the chromosome towards itself, this portion of the chromosome towards itself, and again this centriole will pull both the chromosome towards itself. So there will be the splitting of the centromere. Centromere splits and goes to the opposite poles. So here we have division of this nucleus completely or chromosome. Now the chromatids move into the opposite poles. They have already moved to the opposite poles, connecting to the centrioles. Now slowly and steadily the nuclear envelope develops. These again decondense to form your chromatic material or the chromosome or the DNA, and the organelles, those have disappeared. The protein that has been synthesized in your G2 phase now helps in the preparation of these cell organelles like your nucleolus, your Golgi bodies, endoplasmic reticulum, and etc. Okay, see how the nuclear membrane is formed, and they are forming two daughter cells, but here we need the division of the cell, or the cytoplasm gets to be divided, that is cytokinesis. See how cytokinesis occur in case of animals. Now the nuclear membrane is formed, the cell organelles have been developed, or they are reformed. Now the cell needs to be divided. The cytoplasm of both the portions need to be divided. Okay, from the side a furrow is formed. Okay, in animals a furrow is formed. So this furrow then again develops into the or moves towards the center. So as it is moving towards the center, here the centripetal type of division occurs. Okay, similarly in case of plants, what happens? The cell plate is formed here. Pink color line, cell plate is formed here. This moves outwards towards the cell wall, outwards. So as it is moving outwards, it is centrifugal type of cell cytokinesis or cell division. Fine.